I want to show you how to complete and turn in a lab assignment. This video will help you to know what to do every time a lab report is due. So I wanted to start with, you can see that we're on our Moodle course page, and this is our Bio 112 NT1 section, General Biology 2, and I have gone to the left side where it says Contents, and I've clicked on Chapter 2, Prokaryotes, and I'm going to scroll down. There's your study plan and your quiz, and then this is the link to the lab 2, bacteria and archaea, and this is the actual assignment where you will upload your lab. So all the labs work this way. So I'm going to show you this one, and then I won't keep recording videos for every lab. But this one, you click on the link, and this will come up. And you will go through and hit next and just go through the entire online portion. Read the introduction. For the Did You Know, you have to click on that, and it will usually give you some, a little box with some extra information. But then you click on next and next, and then you'll have a question. And I'm going to deliberately get this wrong because I'm not sure what happens when you get it wrong. But it says cocci bacteria could also be described as. I know the answer is spheres, but I'm just going to put rods. And it says that is incorrect. Please try again. And I guess it just has you continue until you get it correct. But now I'm not going to go through every single thing, but you're going to go all the way through all of this stuff on the left side. And then there's a lab drill where there's some questions that you have to answer. Then you'll get to assignments and procedures, and this is the part that actually contains your experiment. You will click on assignments and procedures, go all the way over to the right and download the lab workbook. Notice that you have two experiments that you have to complete, experiment 1 and experiment 2. So I'm going to download my lab workbook, and this is what I will complete and turn in. A lot of students do not do this. And I end up getting just the data tables and maybe some photographs, but no pre-lab questions. But you'll see here that there are some pre-lab questions. You'll put your answer, like it says, based on the scientific name Streptococcus agalactiae, what morphology would you expect these cells to have? And I'll go ahead and tell you they're going to be round and in chains. So I would put that. I would say round and in chains. And you might even want to put this in a different color. It's always helpful to me. So I got that in red. That helps me when I'm grading to know what's the question and what's your answer. But if you don't do it, it's okay. This is going to be drawings that you complete. And you can print this page out, screenshot it, and turn it in, or you can do the drawing directly, online. But you're going to draw your plates, and I will give you another option as well. Instead of drawing your plates, you can also do you can do a photograph of all of your plates, as long as it's clear, as long as it's very clear. And then you're going to answer questions 2 and 3 about the plates where you grew your bacteria. And then you're going to do experiment 2, your post-lab questions, and then you're going to save this. Make sure you pay attention to how you saved it and where you saved it. And then I'm not going to save it, but you will. And then you are going to, let's go back to our page. You're going to be done with the lab at that point. So you will upload the assignment here. This is not going to look the same to me as it would to you, but you'll click on the actual assignment. It tells you the due date, due January 21st. That will give you not only a grade, a lab report grade, but also an attendance. You will get counted absent if you don't turn it in on time. And you can read this. Click on the link to lab. This is the instructions, which I've already gone over with you. But I want to go all the way to, okay, so it says you will need to complete a lab workbook for both these experiments. Also, this is important, you will take a photograph at the end of both experiments. This is not just for lab 2, it's for all labs. The photograph should include all of your Petri dishes as well as yourself. You should be in the pictures. You can do a selfie, but you have to get kind of good with your camera, or you can have someone else take the picture. But you have to be in the picture, and so does your lab setup at the end of your experiment. And for this lab, you'll have Petri dishes at the end of the experiment. So this is proof to me that you actually did the lab yourself, and that's why I require it. You must have the photographs at the end of both experiments to get full credit. You will have points deducted if you don't turn the photographs in. Sometimes you will actually, like with your dissections, you'll actually have to video record. So this is just a photograph. And then it says this assignment is due on January 21st for attendance and grading purposes. And your page will actually have a submit button, and you will submit. You will upload your lab workbook and you will submit it. Okay, so another thing that I want to show you, there are videos that show you, that actually show you how to prepare your Petri dishes. But there is one thing in the videos that it doesn't show, and I want to make sure that you understand how to do this. So, okay, I'm going to try my best. I don't draw very well, but when you prepare your Petri dishes, each Petri dish has a bottom, and then it has a lid over top of that. Okay, now you can see how terrible I draw. So you've got a top lid that's larger than the bottom lid. And it is the bottom lid that you're going to pour your agarose gel into. So when you first do that, your gel is going to go in here. And so you're going to kind of, as long as the gel covers the bottom, you don't need much. But you're going to cover the bottom with liquid agarose gel, and then that agarose gel is going to harden. Once the agarose gel hardens, you need to flip over your Petri dish so that the top is on the bottom and the bottom is on the top. So, but wait until the agarose gel hardens, otherwise the agarose gel is going to spill. But once it hardens and forms a gel, then flip it over and store it where you've got your bottom end up and your top end on the bottom. Store it like that, because what will happen if you don't is condensation will build up in this lid, and that condensation will end up, the droplets of water will collect and they will fall into your agarose and liquefy it again, and it won't be any good. So make sure that once the gel has hardened that you flip it over, and always store your Petri dishes this way. Always store them with the bottom up and the top down. And so good luck with your lab, and let me know if you have any questions.